The overall goal of this procedure is to visualize Drosophila tissues in situ using fluorescence microscopy following sonication. This is accomplished by first collecting embryonic and larval samples and then fixing them using formaldehyde, heptane, and methanol. The second step is to sonicate the fixed sample in order to remove the cuticle to permit antibody penetration. Next, the sample is immunostained with primary and fluorescent secondary antibodies. The final step is to mount the sample in a glycerol-based anti-fade solution. Ultimately, confocal or epifluorescent microscopy is used to visualize the developing tissue as well as protein expression and localization.
The main advantage of this technique over other methods like tissue dissection is that it allows for in situ visualization of a variety of developing tissues.
Generally, individuals new to this method will struggle because the sonication step is dependent on a number of factors to achieve maximum efficiency, including the proper positioning of the sonicator probe.
Demonstrating the procedure will be Ashley Fidler and Lauren Boulay, two talented undergraduate researchers from my lab.
To begin the experiment, carefully remove embryos collected on an agar plate using a small paintbrush wetted with phosphate buffer Triton X100 or PBTX solution. Next, wipe the sample-laden paintbrush against the interior-facing ridge of a cell strainer. Place a petri dish cover underneath the cell strainer, then rinse the paintbrush and the walls of the cell strainer with a squirt bottle containing PBTX. After the sample has been transferred to the strainer, pour